What's up, everyone? Today I'm going to walk you through variant nomenclature, basically how we describe genetic variants in a standardized way. Before we can get into nomenclature, let's quickly ground ourselves in what we're actually describing. When we talk about genetic variants, we're talking about changes in DNA, which ultimately can affect RNA and then protein. So remember, transcription is the synthesis of RNA from a DNA template, and translation is the synthesis of a protein from an mRNA template. And that matters because variants can be described at multiple levels. Now, who decides how we name these variants? There are actually organizations that standardize this. The main one is the Human Genome Variation Society, or HGVS, which created the system we use today. You'll also see the Hugo Gene Nomenclature Committee, HGNC, which assigns gene names, and the NCBI Reference Sequence Database, which provides reference sequences. And that leads into a really important concept. Every variant is described relative to a reference sequence. The easiest way to think about this is to picture zip codes in an address. A variant without a reference is like an address without a zip code. You might know what the change is, but you don't know where it is. So you'll often see designators like NC, NG, and NM. NC represents the complete chromosome, NG the genomic region, and NM the protein coding transcript. These are database accession numbers referring to specific reference sequences. They designate a specific file in the reference sequence database. In clinical reports, you'll most often work with the NM prefix, which is the coding transcript. An area that can often be confusing is all the letters and dots used to define the type of molecule and coordinate system used in variant description. These tell you what level of biology you're looking at. So the C dot represents coding DNA, N dot non-coding DNA, G dot genomic DNA, M dot mitochondrial DNA, R dot RNA, and P dot protein. So for example, C.76G to C represents the DNA chains, while P.arginine 25 histidine represents a protein change. And typically, labs will report both. These prefixes are essential for ensuring that the location of a genetic variation is unambiguous across different studies and databases. Now let's talk about numbering. For coding DNA, C dot, numbering starts at the A of the ATG start codon. So C.1 equals the A in ATG. From there, numbers increase as you move downstream. If you go upstream before the start codon, you use negative numbers like C.-1, C.-2. If you go past the stop codon, you use an asterisk, so C.*1, C.*2. So this system is really anchored around the start and stop of the coding region. What if the variant is within an intron? Instead of starting fresh, we describe it relative to the nearest exon. So the beginning of the intron is written as the number of the last nucleotide of the preceding exon, a plus sign, and the position in the intron. For example, C.69 plus 1G indicates one base into the intron after the exon. The end of the intron is written as the number of the first nucleotide of the following exon, a minus sign, and the position upstream in the intron, like C.70 minus 1G. This is especially important for splice size variants, which often occur right at these boundaries. Now let's go through the main variant types you'll see. The first and simplest you'll encounter is a substitution. This is when one nucleotide is replaced by another. For example, C.76G to C. This type of variation can lead to missense, synonymous, or nonsense changes, which I'll talk about in more detail later in this presentation. The next type of variant is a deletion. A deletion means that one or more nucleotides are removed. For example, C.7 to 8 del TG denotes the deletion of a T and a G nucleotide from their position 7 and 8 in the sequence. Next, we can have a duplication. A duplication means that a sequence is copied and inserted right next to itself. For example, C.5 dup T. This denotes a duplication of the T nucleotide at position 5. We can also see an insertion. An insertion means that a new sequence is added, but not copied from nearby DNA. For example, C.1228 to 1229 insertion GG. This denotes that the nucleotides GG were inserted between the nucleotides at position 1228 and position 1229. Next, we can see an inversion. This is when a sequence is flipped and reversed. For example, C.5657 to 5660 inversion. This denotes the reversal of nucleotides 5657 to 5660, and it changes an AGGCTGATG to AGGAGTCTG. A more complicated variation you might see is a deletion insertion, also known as an indel. This is when a range of nucleotides are replaced by a sequence from elsewhere in the genome. For example, C.11 to 16 del TGTGCT insertion AA. This denotes the replacement of nucleotides 11 to 16, also known as TGTGCT, by AA. Now let's connect the DNA changes to what actually happens in the protein. When one amino acid changes to a different amino acid because of a single nucleotide change, it's called a missense variation. However, when a single nucleotide change occurs but the resulting amino acid stays the same, it is called a synonymous variation. This is often benign, but not always. Now, a change in DNA that causes a protein to terminate or end its translation earlier than expected because a premature stop codon is created is called a nonsense variation. This often leads to a truncated protein product. When an insertion or deletion occurs that is not divisible by 3 and the reading frame gets shifted, this is called a frame shift variation. It's often very damaging because it changes everything downstream and introduces a premature stop codon. Also, when a variant occurs at exon-intron boundaries, that is called a splice site variant. It can cause exon skipping or intron retention, which can significantly alter the protein. Now let's take a look at some key symbols you might see on a genetic report. A greater than symbol describes a substitution variant. An underscore indicates a range. An asterisk represents a stop codon in protein nomenclature or a downstream region in coding nomenclature. An equal sign represents a synonymous alteration, and a question mark means that the protein change is unknown in protein nomenclature. These are worth knowing cold for exams and clinical work. Sometimes you'll encounter multiple variants. Phasing notation will tell you whether these variants are on the same chromosome or not. A cis variant is represented by both variants within the same brackets, separated by a semicolon. Again, and this represents the same allele. A trans variant is represented by each variant in its own brackets, with a semicolon between the brackets. And again, this is opposite alleles. This can be really important clinically, especially for recessive conditions. One important real-world note in practice is that most labs give you C dot and P dot notation together. And you'll occasionally run into legacy nomenclature, especially with older variants. So always double check databases like ClinVar or HGMD when something looks off. Now let's put this information all together. When you see something like NM underscore 004006.2, C.4375 C to T, you can now break it down. When you ask which transcript, the NM indicates that this is a reference sequence transcript. And the 004006.2 is the unique accession number for the specific transcript variant of the gene in question. In this case, it represents the dystrophin gene. When you ask where in the gene, the C dot specifies that the change is located in the coding DNA sequence. When you ask what nucleotide changed, the 4375C to T indicates that a cytosine nucleotide at position 4375 of the coding sequence underwent a substitution with a thymine nucleotide. And when you ask what happened at the protein level, the original arginine is replaced by a termination codon, creating a premature stop codon. This often causes a truncated dystrophin protein associated with Duchenne or Becker muscular dystrophy. And that's your review. Hit that like and subscribe button for more quick study tips, and I'll see you next time.